I welcome you, my dear students. So now in this video lecture tutorial, we will discuss about the DNA replication in eukaryotes. Already in the class, we have discussed about the importance of DNA replication and when the DNA replication occurs during cell cycle. All those things we had already discussed in the class. Now in this video tutorial, we will discuss about how DNA replicates in eukaryotes. Okay. So first of all, few things we need to know before going to detail. DNA replication is the most fundamental action. Why it is the most fundamental action? Because if you don't understand the DNA replication, you cannot modify, you cannot understand the cell cycle, you cannot modify the genome, you cannot edit the genome, you cannot understand the DNA sequence, you cannot understand the gene, exon, intron concept, all the things, okay, or even the cell division followed by cell cycle. So DNA replication is the most important things. It is a biological process and occurs in all living cells. So it is understood is a biological process and in the living cells also, and it is basis of biological inheritance, is obviously for the biological inheritance. Now there are several processes actually by which DNA replication occurs. Already you have very much familiar with these things like semi-conservative method, conservative method, and dispersive method. So in the semi-conservative method, you can see the two parental DNA strands, and each make a copy of itself. See here, two parental DNA strand, each they make, here is the red one and there is the new one blue color, here is the red one and here is the new one blue color. Okay, each parental strand makes their own new copy, and this is the after first replication cycle. But after the second replication cycle, the two daughter molecules of S comprises one old and one new strand. Note that after two round, two of DNA molecules consist of only the new material, while the other two contains one old and one new strand. See here, this is after the first replication cycle in the semi-conservative model. Both the DNA strands will make a new strand. From this one, they will make similarly from both the strand will be used at the mother strand, and they will replicate and produce this type of DNA double helix. Okay. So in case of conservative, you can see conservative, the terms itself, it means is very conserved because the parental strain in this model, the parental molecule direct synthesis of an entirely new double-stranded molecule. Entirely, this is a very new double-stranded molecule, and this is the existing copy. This is a very new double-stranded molecule, and after the first replication cycle, during second replication cycle, these two new molecules will form, and from here one existing and one new molecule will form. Okay. And in case of dispersive, dispersive means in between the whole genome the replication will be randomly. Here you can see that means each of the strand will act as the replication site randomly. Okay. So randomly between the two daughter molecule, the molecules shown here, the old materials is distributed symmetrically between the two daughter molecule. Symmetrically distributed in the two daughter molecule. Here also you can see. So these are very common DNA replication method, but among all the replication methods, semi-conservative methods is basically